Welcome, I'm Jérémie Perrin, PhD student in bioinformatics at Aix-Marseille University, supervised by both Christine Brun and Olivier Destin. I'll be presenting my work on intracellular signaling models. So what is intracellular signaling? It is a complex network of protein interactions capable of probing, processing, and responding to stimuli from the extracellular environment. We'll be interested in reconstructing this signaling network in order to understand how the cell is able to take decisions. Why do we say decisions? Well, because the cell is capable of responding specifically to a great number of signals, while using only very few intermediary signaling elements to provide a whole class of responses. So, how is it capable of doing so? We'll be interested in the interplay between cell migration and invasion. They are important in both physiological conditions for the immune system, for example, and in pathological conditions, in cancer propagation. A key regulator of both migration and invasion is the Src tyrosine kinase, a first proto-oncogen. We have optogenetically engineered the mutant of the Src protein, opto-Src, and shown by controlling its subcellular localization, we can differentially induce either invadosomes, an evasive phenotype, when opto-Src is free to diffuse in the cytosol, or induce lamellipodia, a migration phenotype, when opto-Src is bonded to the membrane. In each experiment, we measure phosphorylation levels of more than 900 phosphotyrosines before activation, 5 minutes and 20 minutes after the opto-Src activation. Using this data, we will try to understand how signal is propagated inside the cell. So how do we model signaling? In order to consider the full complexity of signaling, and based on a review from Friedman and Perimont in which they advocate for network signaling models, we propose a network signaling model based on network diffusion. We will be able to learn from phosphoproteomics data the contribution of each interaction to the global signal output. So how do we proceed? Well, we use random walk with restart, a network diffusion method. Here you can see the effect in green of diffusing from opto-Src in a network. The greener the protein, the more the protein receives signal. Notice the triangular proteins. The triangular proteins are the proteins which are differentially phosphorylated in our experiment, and for which we have quantitative information of the signal that they receive. So now, can we reconstruct how the information has flown from opto-Src to these triangular proteins? Well, we can do that. We infer the interaction weights, which lead to the measured phosphorylation variations. How do we do this inference? We have shown, it's our theoretical result, that the problem is a linear program. That is, it is of a kind for which we have good optimizers. And so we can solve this problem for very big networks. Once we have the edges computed, we extract paths from source to target. We extract the paths which are significantly biased towards in our experiment by computing empirical p-values through random sampling. Finally, we are able to extract a signaling network. This specifically is the result from applying our method to data from the opto-Src experiment, in the case where opto-Src is bonded to the membrane, which leads to lamellipodia phenotype. You can see the network signaling originating from Src at the center and diffusing towards the green differentially phosphorylated proteins. We can produce signaling networks for both experimental conditions, and we will be able to compare them to hopefully have insights in decision-making capacities of the cell. So those are method work. Well, briefly, we are currently working on a set of in silico experiments to assert the validity of our model and evaluate its predictive capabilities. So both bootstrapping method and applying our method on well-known in vitro data or in silico data, as well as having experimental validation from our partner in Grenoble. Thank you for listening.